Throughout the history of us humans, there have always been a lot of questions with regards to the two sexes, the males and the females. The amount of relationship books out there in bookshops titled something along the lines of Men are from Mars and Women are from Venus clearly indicates that we don't actually know a lot about the opposite sex. Why is this the case, and what are the structural differences in our brains? Don't worry, it's not that we are not intelligent enough to comprehend this. When asked what Stephen Hawking thinks about most, he brilliantly replied, "Women. They are a complete mystery." Although the general public believes that sex is purely based on external genitalia, according to neuroscientist Eric Verlaine from UCLA, there are actually 7 parameters that can be assessed to establish sex, including sex chromosomes, sex-determining genes, brain sex, etc. In the next 4 minutes or so, we're going to focus on the genetic basis of sexual dimorphism in brains of different organisms and their associated functions and behaviors. In humans, the XX pairing represents the female characteristics, whereas the XY pairing represents the male characteristics. But did you know that all of our fetal brains were once in fact unisex until the 8th week after fertilization? The reason behind this is because of the Y-linked SRY gene, which is expressed during sex differentiation in embryogenesis. This sex-determining region Y gene encourages the development of the testes, which will then produce 3 important biomolecules, namely anti-Mullerian hormone, insulin-like peptide 3, and testosterone. Testosterone would then turn this unisex fetal brain to male by killing off some brain cells in a communication center and growing more brain cells in a sex and aggression center, such as the amygdala. In the absence of these 3 biomolecules, however, the unisex brain would then develop into a fetal girl's brain. Considering the timeline of the evolutionary history of life, it is no surprise that we are only one of many species on this planet that have sexual dimorphism in the brain. Many neuroscientists and geneticists' favorite insect, Drosophila melanogaster, also have brain sex differences and intriguing behaviors associated with them. In male Hawaiian fruit flies, it has been shown by Japanese scientists such as Daisuke Yamamoto that they have a bigger DA1 glomerulus than the females, which is a region of densely packed neuropils on the antenna lobe of the fruit fly's brain that receives input from olfactory receptor neurons, ORNs, on the antenna. One type of ORN expresses an odorant receptor called OR67D, which is a receptor for the volatile pheromone fascinyl acetate, CVA, and it's coded for by a gene called LUSH. According to a paper published in Nature by Dr. Kurtovich in 2007, if this gene is mutated, male fruit flies that are mutant for OR67D start courting other males, whereas females that lack OR67D become less receptive towards males courting them. It is fascinating to know how a change in the DNA sequence of a gene could lead to such drastic changes in sexual behaviors shown by fruit flies. In addition to our findings, there was also research done in the US on the sex differences in the cerebellum and the frontal cortex in mice, which related to a gene called Kelv1, as well as an experiment done in Sweden on sexual dimorphism in sex-biased gene expression in 2 species of songbirds. In fact, there haven't been a lot of papers published about genetics and brain sex differences in the scientific community, yet scientists in this particular field are truly remarkable and admirable in that they've helped shape the modern knowledge about the relationship between genetics and neuroscience, as well as helping us to understand more about the opposite sex, both at the social and at the molecular level.